Hi, my name is Casey Owens, and this is my Unit 1 presentation in Applied Genetics. It is the 4th of July, 2024, and this is my 100th take because I keep making a video that's way too long, so hopefully I will be concise here. So the article covers what are called SITs, or sterile insect techniques, as they relate to genetic biology. So there was a little bit of background in the 1930s and 1940s, sterile insect techniques, which are used in the management of insect pests as they relate to agricultural crops, they were conceived of independently by a couple of different scientists, and some thought to crossbreed hybrid strains to affect sterility in the populations and then release those into the wild. Others thought radiation was the way to go. So currently, radiation is what's being used, and the way that works is insect populations that are grown in captivity are dosed with radiation, hopefully eliciting chromosomal damage that affects sterility. And those sterile insects are released into the wild, compete for mates with wild insects, and suppress the insect population. So this isn't a perfect system. There's a lot of collateral damage when you dose an organism with radiation. So there's a huge opportunity for genetics-based biology to step in here. Systems research is being done on raising males that are homozygous for a dominant lethal allele and releasing them into the wild population. In captivity, they're fed a diet that represses the expression of the dominant lethal allele so that they can be raised to reproductive age and released into the wild where they compete for mates. Obviously, because it's a dominant lethal allele and they're homozygous, their progeny all inherit one of the alleles. It's dominant, it's lethal, the progeny are non-viable. This suppresses the insect population. There's a further opportunity. What's great about genetics-based biology is that we can be really specific for a dominant female-specific allele. So the way this would work is very similar. Is that mosquitoes are bred in captivity. It's a dominant female-specific allele. They're taken off the repressor diet. The females die off. The males that are homozygous for a female-specific lethal allele are released into the wild. All of their female progeny die because they inherit a female-specific dominant allele, and male progeny also inherit this allele, so their female progeny die as well. So these can be much more fine-tuned approaches to pest insect population control. So there's also, the authors talk about genetically modified crops, or BT crops. So these are agricultural crops that have been genetically modified to have insecticidal toxins from a bacteria that obviously kill pest insect populations. So doing this on its own doesn't work and is not viable, because eventually resistance to the BT toxins will evolve in the population and quickly be selected for. And once the resistant allele is the more frequent allele in the population, the entire investment in the BT crops is null, and you have to start all over again. So what's been developed is what's called a high-dose refuge system. And in the high-dose refuge system, there are two parts. One is the high dose, which means there's a high enough dose of insecticidal toxins from the BT in the crops to kill even heterozygotes for resistance. So that means individuals who have an S and an R allele susceptible to the toxins and resistance to the allele do not have enough resistance to survive ingestion of the BT toxins, and they will die. And the refuge system addresses the selection for resistance. So the refuge, it would be a nearby crop field. It could be the same or a different crop that's not genetically modified to have BT toxin. It's a safe haven, so to speak, so that individuals with susceptible alleles can go and reproduce and continue to keep the susceptible allele, the more dominant one, the more frequent allele appearing in the population, so that we don't select for resistance so quickly. Unfortunately, eventually resistance is selected for, and it becomes a more frequent allele in the population. So imagine now combining the genetics-based approach to sterile insect techniques with this high-dose refuge technology. And that's what's being explored and implemented in some places right now. So in this ideal situation, males that are homozygous both for susceptibility to the Bt toxin and for the dominant female-specific lethal allele are released into the wild population where they compete for mates. And here's what happens. They're homozygous for susceptibility. So not only are they susceptible to the Bt toxins, but their progeny inherits susceptibility to the Bt toxins, making them at least heterozygous for resistance. And with the high dose of the Bt toxins in the crops, they die. They're also homozygous for the female-specific lethal allele that's dominant. So all of their female progeny will die, and all of their progeny will inherit a dominant female-specific lethal allele. So when this technology, genetics-based technology of genetic modified insects, is combined with the high-dose refuge areas, this is very successful. Models have shown that even modest release ratios, meaning the ratio of genetically modified insects to wild-type insects, would be very successful in a program such as this. And in fact, they could be used remedially to correct for high-dose refuge systems in which the resistance allele is already the more frequent allele. So it's really suppressing or eradicating those insect pest populations. So basically, the authors were just talking about the opportunity there is for the genetics-based approach to sterile insect techniques. It's not just that easy. This is a very interdisciplinary approach and endeavor that requires, obviously, knowledge of molecular and genetic biology, behavioral ecology, economics, and then there's a social, ethic, and legal aspect to it. But the authors just wanted to share the cutting edge here. My personal opinion is I didn't realize that genetically modified organisms were being used for this purpose. I think it's great. I don't really see a downside to it. I hate mosquitoes, so I think this is wonderful. This is a great opportunity for genetics-based genetic engineering to maybe solve a hunger problem and really save billions of dollars every year in pest management and lost crops. Thank you very much.